So, good morning everyone. I am Ellen V. Joan S. Cabrera, and let's discuss Lesson 4 about classification of lipids and their reactions. So, classification of lipids. Lipids can be classified according to their hydrolysis products and according to similarities in their molecular structures. So, three major subclasses are recognized. These are the simple lipids, the fats and oils, and the waxes. So, let's dwell first on simple lipids. First is the fats and oils, which yield fatty acids and glycerol upon hydrolysis. So think of a Lego blocks. And when you take them apart, muna sila through a process called hydrolysis, and when broken down, muna sila mo kanog do kabuok si glycerol and the fatty acids. So si fatty acids is the small pieces of Lego blocks, while si glycerol muna siya the base or the platform that holds the pieces together. And so fats and oils when broken down, wag ni sila duha kabuk nga magbahin, si fatty acids and si glycerol. And so, let's move forward to waxes. Waxes which yield fatty acids and long-chain alcohols upon hydrolysis. Let's dwell a little bit about waxes because this will be discussed further in the next video. So waxes are compound that found in nature like beeswaxes, plants waxes, and when broken down through a process called hydrolysis. And when waxes are broken down, babahin na po ni sila do kabuo. Si fatty acids and si long chain alcohol. And so when waxes are broken down through hydrolysis, they produce fatty acids and the long chain alcohol. So fatty acids are important for our energy and cell structure, whilst the long chain alcohol, which has various functions in organisms. And so let's go back to fats and oils. Fats and oils are known as lipids, which are important molecules in our energy storage and cellular structure. They are made up of building blocks called fatty acids and glycerol. Fats are usually solid in temperature, while oils are liquid, and this difference is due to their chemical structure. So fats are mostly saturated fatty acids, while the oil is unsaturated fatty acids. And fatty acids is very important to our energy storage, and lipids serve as a concentrated source of energy and important to our functions and structure in our cell membranes, and plays a role in signaling pathways and hormone production. And that's all. Thank you.
So, good morning everyone. This is the continuation of our report in Lesson 4, Classification of Lipids under Reaction. So first, what is lipids? So, lipids are fatty compounds that perform a variety of functions in our body. They're part of your cell's membrane and help control what goes in and out of our cells. They help with moving and strong energy, absorbing vitamins and making hormones, but too much lipids is harmful. So there are three classification of lipids. The first is simple lipids, second is compound lipids, and the last is derived lipids. So since simple lipids has already discussed, so I am going to discuss the compound lipids. So what is compound lipids? So compound lipids, this includes the fatty acids, the alcohols, the monoglycerides, and the diglycerides, and steroids, terpenes, and carotenoids. The most common among them are steroids, terpenes, and carotenoids. So, so compound lipids has as phospholipids, which yield fatty acids, glycerol, amino alcohol, sphingosine, phosphoric acid, and nitrogen containing alcohol upon hydrolysis. So hydrolysis, it is the one from the ancient Greek, Latin, from the word hydro means water, and this is means to break down. So they commonly use water to break down the chemical reactions. So they may be glycerophospholipids or sphingospholipids depending upon the alcohol group present or glycerol. Second is the glycolipids, which yield fatty acids, sphingosines or glycerol and carbohydrate upon hydrolysis. They may also glyceroglycolipids or sphingoglycolipids, depending upon the alcohol group present, glycerol or sphingosine. So glycerophospholipids, membrane lipids. So 1 to diacylglycerol, the fatty acid with a phosphate group at position, essential components of cell membranes and other circular structures. The parent molecule is phosphatic acid. You have additional compounds enterified to the phosphate group. So below is the structure of the phosphatidic acid. So this is the structure of the phosphatic acid. Then phosphati so below are the example of glycerophospholipids like phosphati diclolene or lecithin and phosphati nolamanin, phosphati dilitanolamine, common membrane constituents. So these are the structures, examples of these glyphos. So that's the end of my topic.
Good morning, ma'am. I am Jane Ibo, Nick's reporter. My topic is all about sphingolipids membrane lipids brain. Sphingolipids membrane lipids brain. Sphingolipids are typically found in brain tissues. Example, brain lipids. Based upon sphingosine or dihydrosphingosine, an amino acid. Sphingosine is a rare in plants and animals, while sphingolipids are common. Simply, sphingolipids are ceramides. Sphingosine plus N-lin fatty acid equals ceramides. Sphingosine HO3 CH double bond CH single bond CH2 12 single bond CH3 fat 2CH single bond and COH 1CH2OX fatty acid. Sphingolipids general structures. Sphingolipids, the first their carbons at the polar end of sphingosine are analogous to the three carbons of glycemials in mophospholipids. The amino acid group at CG for a fatty acid in amine linkage. The fatty acid is usually serrated on most serrated, which 16, 18, 22, 24 carbon atoms. Ceramides is planet carbon. Fourth biggest group, polar head plus 2 non-polar tail. No glycerol. Majority no phosphate groups. Extra sphingosine is structures 1 sphingosine 1 Fa plus head. H2C single band C, single band C, single H. H to C, single ni function siya OH. Ang C, ni function siya og H. C, ni function na siya OH. Sa ubos, ang C, ni function na siya H3N+. Next, C, ni function siya CH. CH, single band HC, single band HCH212, single band CH3, muna siya sphingosine. Next, H to C, single band C, single band C, single band H. H to C, single band OH, C, single band H, C, single band OH. Single band C, H3N plus, single band C, CH2, single band CH2, single band CH212, single band CH3, meron siya dehydrospingosine. Sphingolipids types. Name of sphingolipids. Name of X, formula of X. Ceramide, negative H, sphingomyelin, phosphocholine, P, single O, single CH2, single band CH2, single band N plus CH3, close parenthesis 3. Neutral glycolipids, leukoacylceramide, locus, H, single band OH, tayo na sa circle, H, O, OH, H, sa gawa CH2, OH, sa ubos, H, OH, H. Lactosylceramide, globoside, T3 or tetrasaccharide, GLC, GAL. Ganglioside, GM2 complex, oligosaccharide, GLE, GAL, GAINAC. Ang GAL na siya i-function na NEU54C. Ganglioside membrane lipid signaling. Ganglioside are complex sphingolipids. Ceramide plus 3 or more sugar including 1 sialic acid. Limit abundance K, tissues specific signaling molecules. GM1 D-galactose, GM2 N-acetyl D-galactosamine, GM3 D-galactose D-glucose, A N-acetate acylic acid, stearic acid, and sphingosine. The sphingosine backbone has a long chain of carbon atoms which as hydroxyl groups on one end of an amino groups bonded to the second carbon atoms from the end. The fatty acid is shown bounded to the amino group of the sphingosine backbone by an amide band. The amide band of shown as a black line between the nitrogen atom N in the amino group and carbonyl carbon atom. C double band O in the fatty acid. Sphingolipids are important for cell structures and function. They help to found the cell membrane which protect the cell and control what enter and leaves the cell. Sphingolipids are also involved in cell signaling, which is how cells communicate with each other. Example of sphingolipids are ceramide, which is simplest types of sphingolipids. Ceramides are made of sphingosine and a fatty acid. They are found in cell membrane and play role cells signaling and cell death. Ganglioside example. Ganglioside determine blood types. O, A, and P antigens that give rise to blood types are ganglioside. Polar hedge groups of these gangliosides difference electrons micrograph of site outer membrane ganglioside. Sphingosine, ceramide, fatty acid, O antigen, A antigen, P antigen. Blood types are determined by the presence or absence of certain carbohydrates called antigens on surface of red blood cells. These antigens are parts of molecules called glycoproteins and glycolipids, which are produced by enzymes that attach carbohydrates to proteins or lipids. There are two main types of antigen, A and B, and a third types O, which is the absence of either A or B antigens. The presence of A and B antigens are determined by the single genes, which three alleles A, B, and O. People secrete two or these alleles, one from each parent. The A and B alleles code for the enzymes that attach specific carbohydrates to precursor molecules. The O alleles does not code for any functional enzymes. So individuals with two O alleles have no A or B antigens.
Good day everyone, I am Josh P. Baconga. Triglycerols or triglycerides, energy storage lipids. One fatty acid and glycerol is called monoacylglycerol, and two fatty acids and glycerol is diacylglycerol. Three fatty acids and glycerol is triacylglycerol, and this glycerol and tristerine is a simple triaglycerol, and myristic palmitoleic steric is a mixed triaglycerol. Triacylglycerol, energy storage lipids, it is a most abundant form of fatty acid, not part of biological membrane. It major energy reserve in animal. Energy yield from burning 37 kg or grams as compared to 16 kg for carbohydrates. Example sugar. Here stored metabolic fuel in a 70 kg male. In energy constituent fat, protein, glycogen, glucose, and in kg is here in fat 37 and protein is 17, and glycogen or muscle is 16, glycogen in liver 16, and glucose is 16. And here fat and mass is 15,000, and in protein muscle is 6,000. In glycogen muscle 120, and glycogen liver 70, and glucose is 20. Here in energy or kJ is fat 555,000, and protein or muscle is 102,000, and glycogen muscle is 1920, and glycogen liver is 1120. Glucose is glucose is 320, in total of 660,000 and 360. Good day everyone. I am Josh P. Baconga, BSA 2 Animal Science. Waxes are lipids that consist of esters formed between a long-chain alcohol, typically monohydroxy, and fatty acid. Waxes are composed of a long-chain alcohol, which is usually monohydroxy, meaning it contains hydroxyl group, and a fatty acid. Through the esterification reaction, the hydroxyl group of the alcohol reacts with the carboxyl group of the fatty acid, resulting in the formation of an ester linkage. This ester bond is what gives waxes their characteristic structure and properties. Due to their unique composition, waxes often exhibit properties such as water repellency, low solubility in water, and high melting point. These properties make waxes suitable for various biological functions, including providing waterproofing and protection for plants, insects, and animals. The acids and alcohol normally found in waxes have chains of the order of 12 to 34 carbon atoms in length. Waxes are lipid compounds characterized by their esterification of long-chain fatty acids with long-chain alcohols. The acids and alcohols found in waxes typically have carbon chains ranging from 12 to 24 atoms in length. This range encompasses a variety of molecule sizes, allowing waxes to exhibit diverse physical properties such as rigidity, melting point, and water repellency. Waxes are highly insoluble esters of chain monohydroxy alcohol. It consists of long-chain monohydroxy alcohols, typically paired with fatty acids. This chemical structure renders waxes resistant to dissolution in water, imparting them with hydrophobic properties. Due to this insolubility, waxes serve essential functions in waterproofing, protection against environmental factors, and structural support in organisms ranging from plant to animals. Their stability is impermeability make them invaluable to diverse applications, including in cosmetics, pharmaceuticals, and industrial coatings. Waxes are esters of fatty acids of high molecular weight with alcohol except glycerol. Waxes characterized as esters formed from the combination of high molecular weight fatty acids and alcohols. excluding glycerol. This structural composition is distinguishes waxes from other lipids like triglycerides. The absence of glycerol in wax molecules contributes to their unique properties, including low water solubility and high melting points. These traits make waxes valuable for functions such as waterproofing, protective coatings, and structural support in biological systems, as well as various industrial applications. So here next, waxes have higher melting point than neutral fats. Waxes typically have higher melting points compared to neutral fats due to differences in their molecular structure. While both waxes and neutral fats are esters of fatty acids and alcohols, waxes have no longer and more complex carbon chains in both the fatty acid and alcohol components. So next, sebaceous glands and mammalian skins secrete waxy sebum which acts as lubricant to keep hair and skin soft. These sebaceous glands in mammalian skin produce the secrete substance called sebum. So it's called sebum, which serves several important functions. Sebum is a waxy oily substance composed primarily of triglycerides, fatty acids, wax esters, squalene, and cholesterol. One of the primary roles of sebum is to lubricate the skin and hair, keeping them soft and supple. When sebum is secreted onto the skin's surface, it forms a protective film that helps prevent water loss and maintain hydration. This lubricating action also helps to prevent friction between the skin and hair, reducing the risk of irritation and damage. So, next, the sebum secreted by skin glands of woolly mammals is commonly called lanolin or wool oil. Lanolin, also known as wool oil, serves as a valuable natural resources with a wide range of applications both animal care and human skin care. Its protective, moisturizing, and soothing properties make it a sought of after ingredient and cosmetics and personal care industry. Next, ceruminous and sebaceous glands of the skin that covers the external ear passage in mammals secrete earwax. Earwax produced by the ceruminous and sebaceous glands of the external auditory canal serves a natural protective barrier, lubricant, and antimicrobial agent. It helps to maintain the health and integrity of the ear. So next, spermaceta, a wax found in skull of whale and dolphin. Spermaceta is a unique wax-like substance found in the spermaceta organ located in the heads of the certain species of whale, including sperm whale and species of dolphins. The spermaceta organ contains a large reservoir of spermaceta oil, which solidifies into a waxy substance at lower temperatures. So, and here next, the honeybees constructed their beehives with bee wax, myrivyl palmitate, secreted by their abdominal glands. So, honeybees constructed their intricate beehives using beeswax, which is primarily composed of myricyl palmitate, a wax ester. Beeswax is produced by special glands located on the abdomens of the worker bees. These glands secrete a liquid wax that hardens upon contact with air, forming characteristics hexagonal cells of the honeycomb structure. Paraffin wax is obtained from petroleum. Candles are made of paraffin wax and stearic acid. Paraffin wax derived from petroleum is widely used material in candle making due to its affordability, availability, and ease of use. It is type of hydrocarbon wax composed primarily of saturated hydrocarbons, which give it a relatively low melting point and a firm yet pliable texture. Carnauba. Carnauba wax, myrisal cerotate, obtains from leaves of Carnauba palm. Carnauba wax is a valuable natural resource with a wide range of industrial applications, prized for its hardness, high melting point, and glossy finish. Its sustainable and renewable source derived from the leaves of the Carnauba palm tree makes it an attractive alternative to synthetic waxes in various manufacturing processes. Uh, waxes are esters of fatty acid and fatty alcohol. Waxes are indeed esters formed from the reaction between fatty acid and fatty alcohol. This esterification process involves the bonding of the carboxyl group and fatty acid with the hydroxyl group of fatty alcohol, resulting in a formation of an ester linkage. Uh, Fatty acid plus fatty alcohol plus wax ester plus water fatty acid and fatty alcohol plus wax ester and water. In this reaction, the fatty acid acts as the acid component while the fatty alcohol serves as the alcohol component. The ester linkage formed between two components gives waxes their characteristic chemical structure and physical properties. Insoluble and water repellent. Insolubility in water means that waxes do not dissolve in water. Instead, they form a separate paste, often floating on the surface or remaining as solid particles within water. This property makes waxes useful in various applications where water resistance or barrier properties are needed. And that's all, and thank you!
Derived lipids. Hydrolysis product of simple and compound lipids is called derived lipids. They include fatty acid, glycerol, sphingosine, and steroids derivatives. Lipids are a class of organic compounds that are insoluble in water but soluble in organic solvent. They are a vital part of the diet of most heterotrophs, including human, and are one of the main types of molecules found in all living organisms. There are three major types of lipids: simple lipids, compound lipids, and derived lipids. So number one, the simple lipids. These are esters of fatty acids with various alcohol. For example, fats and esters of fatty acids with glycerol. Number two, compound lipids. These are esters of fatty acids with an alcohol, and in addition, they contain groups such as phosphoric acid, sugar, nitrogenous, and bases, or etc. Phospholipids and glycolipids are examples of compound lipids. So number three, derived lipids. These are the substances derived from the hydrolysis of the above two types of lipids. They include fatty acid, glycerol, steroids, and other similar substances. Hydrolysis is a chemical reaction in which water is used to break down a compound. During the compound of lipids, water breaks the ester bond, producing glycerol and fatty acids. So, fatty acids are carbocyclic acid with long aliphatic chain. They can be either saturated or unsaturated. Glycerol is a simple polyol compound. It is a colorless, odorless, viscous liquid that is sweet-tasting and non-toxic. Sphingosine is an amino alcohol with a long unsaturated hydrocarbon chain. Sphingosine is a component of some types of sphingolipids, which are a class of lipids found in biological membrane. Steroids derivatives are a type of lipids molecule that includes molecules like cholesterol and steroids hormone. Steroids derivatives are phenanthrene structures that are quite different from lipids made up of fatty acid. So, steroids derivatives are compounds that share a common phenanthrene structure, which consists of four fused rings. These compounds are distinct from lipids, which are molecules made up of fatty acids. Steroids play an essential role in the body, serving as hormones, cholesterol, and bile acids, among other functions. Lipids, on the other hand, are a broad category of molecules that include fat, oils, waxes, and other related compounds, serving as energy storage, structural components, and signaling molecules. They are produced by hydrolysis of the above two groups, or they represent in association with them in nature. These include one fatty acid, both saturated and unsaturated, two alcohols, A. Glycerol, B. Sterols, and other steroid including vitamin D, C. Alcohols other than glycerol and sterol, D. Fatty aldehydes. Derived lipids are substances that are produced by the hydrolysis of simple and compound lipids. They are present in association with them in nature. They are typically less complex than simple and compound lipids and include substances such as fatty acids and alcohol. Fatty acids are organic molecules that consist of hydrocarbon chain and carboxylic acid group at one end. They are classified into main categories: saturated fatty acids and unsaturated fatty acids. 1. Saturated fatty acids. Saturated fatty acids contain only single bonds between carbon atoms in their hydrocarbon chain. They are typically solid at room temperature. Common food sources include animal fat, coconut oil, and palm oil. Example include palmitic acid and stearic acid. 2. Unsaturated fatty acid. Unsaturated fatty acid contain one or more double bonds in their hydrocarbon chain. They can be further classified into monounsaturated and polyunsaturated fatty acid. Monounsaturated fatty acids have one double bond, while polyunsaturated fatty acids have two or more double bonds. They are typically liquid at room temperature. Common food sources include olive oil, avocados, and nuts. Example include oleic acid, monounsaturated, and linoleic acid, and polyunsaturated. Glycerol, glycerol, also known as glycerine or glycerine, is a simple polyol compound. It is a colorless, odorless, viscous liquid that is sweet-tasting and non-toxic. It is widely used in food industry such a sweetener and humectant and a pharmaceutical formation. Sterols and other steroids, including vitamin D. Sterols, also known as steroid alcohol, are a subgroup of steroids with a hydroxyl group in the 3 position of the ring. They are important cell membrane components and have various biological functions. Number 1. Cholesterol. This is the most common type of sterol and is crucial for all animal life. It's used to build the structure and fluidity of cell membranes and is a precursor of steroid hormones, bile acids, and vitamin D. Petosterol. These are plant sterols similar to cholesterol, which occur naturally in plants and vary only in carbon side chain and or presence or absence of a double band. Include cetosterol, cam, campesterol, and stigmasterol. Number 3, ergosterol. This is a sterol found in the cell membranes of fungi and protozoa, serving many of the same functions that cholesterol serves in the animal cells. Here are some examples of steroids. Number 1, corticosteroid. These are a class of steroid hormones that are produced in the adrenal cortex. Example include cortisol, which helps regulate metabolism and the body's response to stress, and aldosterone, which controls blood pressure. 6. Steroids. These are steroids that interact with vertebrates' androgen or estrogen receptors. Example include testosterone, estrogen, and progesterone. 3. Anabolic steroids. These are synthetic variants of the male sex hormone testosterone. They are used to promote muscles and bone mass growth. C. Fatty aldehydes are organic compounds that contains a terminal aldehyde group at the end of a long aliphatic carbon chain. They are intermediates in various biological pathways and play important role in cellular processes. Steroids comes under the category of lipids. Steroids are diploid lipids. Composition contain a characteristic arrangement of 3 cyclohexane ring, 1 cyclopentane ring, a total of 17 carbon atoms, and 4 fused carbon ring. Steroids do not contain alcohol and fatty acid. Testosterone is a well-known and important hormone in the human body. It is a steroid hormone from the androgen group and is primarily produced in the testicles in males and in smaller amounts in the ovaries in females. Testosterone plays the crucial role in the development of male reproductive tissues and promotes secondary sexual characteristics such as increased muscles and bone mass, facial and body hair growth, and deepening of the voice. Cholesterol is a type of lipid that is crucial for many biological functions. It acts as a substance that found in all cells of the body has a complex structure. Number 1, biomembrane. Cholesterol is an essential component of cell membranes. Contributing to their fluidity and integrity, it helps maintain both the physical and functional properties of the membrane. Number 2, nerve tissue. In the nervous system, cholesterol is a critical component from the formation and function of myelin, which insulates nerve fiber and enhances the speed of nerve impulse transmission. Number 3, adrenocortical and sex hormone. Cholesterol is the precursor molecule for the synthesis and steroid hormones, including adrenocortical hormones or cortisol, aldosterone, and sex hormones estrogen, progesterone, and testosterone. Vitamin D. It is also a precursor for the synthesis of vitamin D, a hormone that is essential for bone health and immune function. Number 5, bile acid. Cholesterol is used by the liver to produce bile acid, which are necessary to digestion and absorption of dietary fats. Cholesterol is based on cyclopentanoperhydroxyphenanthine ring, which is a fused ring system consisting of 3 cyclohexane rings and 1 cyclopentane ring. The structure also includes a hydrocarbon tail and hydroxyl groups at one end of the molecule. These complex structures allows cholesterol to interact with both and fatty acid chains and the polar heads of phospholipids in the biomembrane, thus playing a vital role in maintaining the membrane's properties.
Good day, everyone. I am Angela Nicole Allen Tulan, and I am going to talk about terpenes and other lipids. Terpenes are abundant in plants. They are built from 5 carbon isoprene units and do not contain fatty acids. Terpenes have different structures. One is monoterpene. It contains 2 isoprene units. Next is the sesquiterpenes, which contains 3 isoprene units. Next is the diterpenes, which contains 4 isoprene units. And lastly is the triterpenes, that which contains 6 isoprene units. They are also generally assembled by a head-to-tail linkage of isoprene units. This is an example of head-to-tail linkage of isoprene units. Now we were going to talk about lipids. So Bloor, 1943, classified lipids into 3 types. We have simple or homolipids, compound or heterolipids, and derived lipids. Simple lipids or homolipids are esters of fatty acids with alcohol. Examples of these are fats, oils, and waxes. Second is the compound lipids or heterolipids. These two components, lipids and non-lipid. Examples are phospholipids, glycolipids, lipoproteins, and sulfolipids. Next is derived lipids. These are derived from simple and compound lipids. Examples are fatty acids, steroids, glycerol, prostaglandins, leukotrienes, terpenes, dolicol, MAG, DAG, fat-soluble vitamins, and ketone bodies. We have different reactions of lipids. One is the hydrolysis of triglycerols. Tri- Triglycerols, like any other esters, react with water to form their carboxylic acid and alcohol, alcohol, a process known as hydrolysis. Second is saponification. When triglycerides are hydrolyzed or saponified by alkalis, glycerol plus the salts of fatty acids are produced. Generally, the sodium or potassium salts are obtained, which are termed soaps. Third is the hydrogenation. The carbon double bonds in unsaturated fatty acids can be hydrogenated by reacting with hydrogen to produce saturated fatty acids. Fourth is halogenation. Unsaturated fatty acids, whether they are free or combined as esters in fats and oils, react with halogens by addition at the double bonds. The reaction results in the decolorization of the halogen solution. Fifth is the rancidity. The term rancid is applied to any fat or oil that develops a disagreeable odor. Hydrolysis and oxidation reactions are responsible for causing rancidity. Oxidative rancidity occurs in triglycerols containing unsaturated fatty acids. This reaction occurs more quickly in the presence of heat and moisture. That's all, ma'am. Thank you.